Thank you all again for joining today's Knowledge Portal webinar. We are looking forward to our jam-packed schedule, so let's get started. First, I want to give an update on a couple events that we're going to be attending. And the first is the American Society of Bone and Mineral Research 2025, which is going to be in Seattle in September. So we will be having a booth that will be represented. So come check us out at booth 124. Here are the dates and times that we are going to be available for the booth. Please feel free to stop by if you have any questions in terms of research or personalized demonstrations. We'd be happy to do that through there. So we look forward to seeing you there, and please reach out to me if you're interested in a personalized demonstration. We will also be at the American Society of Human Genetics in 2025. This is going to be in Boston in October. We are going to have a booth, and we will also be hosting an ancillary session. Please feel free to stop by for this as well throughout these days. The ancillary session is going to be October 15th from 11:45 a.m. to 1:15 p.m. in the Convention Center. We will have more details on this event as we get closer to, but we'll be sharing some pretty exciting research that's going on in the CMDKP and A2F space. One other event that I just want to highlight for another funding mechanism that we have is through the Common Fund Data Ecosystem. And our next webinar is going to be Friday, August 29th from 1 to 2 p.m. and will feature xRNA, specifically mapping the extracellular RNA landscape, a resource for minimally invasive biomarker research. So please feel free to join us for that as well. If you're unable to attend, we will have the webinar recording available afterwards. And just looking ahead to some of our future meetings, today is the August 14th webinar. September 11th will be our focus group. October 9th will be the next webinar. November 13th will be our focus group, and December 11th will be the last webinar of 2025. So like I said, thank you all for joining today. I am going to have Noel start us off for this session. And I mentioned this before, but please feel free to use both the chat and question and answer throughout our time today.
Okay. Hi, everyone. Hope you all can hear me. I'm just going to get everything set up and then I'll switch over to my slides in just 1 second. Of course, nothing's working right now. Isn't that awesome how that happens?
It's okay.
I think we have plenty of time, so just give me a second here.
Definitely.
There we are. Does that look like my slides?
Yes, yeah.
All right, super. Okay, so we have 3 presenters today, and I'm really excited to talk about this because this has been a labor of love for many, many years now for several of us on the team, particularly Doug Keel, who I'm imagining is joining. But the Musculoskeletal KP has been around since 2019, I think, officially. And it really was started by a grassroots effort by Douglas Keel, who's a member of, a Broad Associate member who we got to know through, he was using the Type 2 Diabetes Knowledge Portal. He was like, you know what? Musculoskeletal traits are not well represented on resources in general. And he's like, I really would like a place where I can contribute my data. So this started way back when other disease areas were doing similar things. And this resource was like, had a little seed funding by a wonderful organization called the IFMRS, who helped us actually aggregate data that was existing at the time for musculoskeletal disease and make a resource that has the same tools and underlying architecture and infrastructure as our Type 2 Diabetes Knowledge Portal and then the CMDKP. But to really sustain, maintain, and understand that community, we wanted to find a mechanism to support this long term. So luckily, NIAMS gave us the faith and trust to support this resource for the next 4 years. Just got awarded to do it recently, to take it from what they call an early stage knowledge base, which has, some rudimentary functionality, some good data, some high potential ideally, to convert it to an established knowledge base. And particularly for musculoskeletal disease, I think we can imagine the challenge of musculoskeletal disease. We don't often think about it as, a disease we will want to study because, things like Alzheimer's are more in our forefront when it comes to common disease or things that are more like debilitating required treatments. But if you imagine, it's a global threat to our aging population, right? We all age and our bones deteriorate, particularly women, and also on top of it, muscle strength deteriorates. And this also contributes to a whole other host of conditions. It also minimizes our movement as humans, right? So the cost annually to this for this industry is over 300 billion and rising, right? So it's a high cost to our medical field and to people's lives, right? What's interesting is there is no disease-remitting treatments, particularly for OA and muscle strength as people age. So it gets worse and worse. So it's a very chronic condition. But what's good is we know that human genetic studies help lead human targets for human treatments and diseases, right? We know this through a lot of the work that we all have done in GWAS for other disease areas. We also know that many of the GWAS associations that have come out from musculoskeletal disease actually have led to some therapeutic targets. So that's great news. But for an example, for like bone mineral density, there are 500 and counting loci for associated, genome-wide significant loci associated with bone mineral density. That's a lot. No mechanisms are clear for that. And this is the case for many diseases. But the interesting challenge with musculoskeletal disease is it's lagging. It's lagging particularly not necessarily in the GWAS space as much, but in the layering of epigenomic, transcriptomic, functional assays integrated with the genomics to make sense of it all to help produce mechanisms. So resources for that. There's lots of research going on, but there aren't many resources to pull that all together. In fact, there's no existing one other than our early knowledge base. And also, we really want to invite basic biologists. We have a small but sort of dedicated following, but that's really in the space of GWAS, people who are taking, a specific gene and trying to move it into translation. But we want to invite cellular biologists, mechanistic biologists, animal model biologists to help use this to translate to human so that we can bring that information to the genetics and help make a resource that's more holistic. So that's really the goal of the established knowledge base. I'm going to talk a little bit more about what we're going to do at the end. But since we haven't talked about the MSKKP, and you probably haven't used it in a while, we thought today would be really nice just to remind you of what's in there now, where we are, how the tools can be navigated. You probably know this if you've used any of our portals, so some of this may be new to some of you. But to you who study musculoskeletal disease, this may be valuable because you may not know it was there. So with that, I'm going to turn over to MacKenzie, who's going to walk us through a demo, and then we have a little bit from me again, where you'll hear a little bit about what we're planning, and then we'll end up with some actual data that's coming forth in September for the MSKKP. So let me stop and turn it over to MacKenzie, unless there's any questions, of course.
All right, thank you. Okay, so Noel did a really great introduction of what we're planning on showcasing today. And I think a big part of what I'm hoping that you'll learn is a little bit more about our current MSKKP site. And then we'll go into what's coming up in our future plans. And then I will share how you can work with us to improve the site long term. Your valuable feedback will help us as we transform this from the current stage that we're at to that established knowledge base, as Noel had mentioned. So this is the first resource of its kind that really allows researchers who are interested in both this disease and associated diseases to get all of the data in one centralized location. And we're hoping that you'll be able to help us formulate what this will look like moving forward so that we can understand what is best for the researchers who use this site. So our website here, this is the homepage. If you head to mskkp.org, this will take you to our site. But I'm going to walk you through first some of the data that's in here, and then go through some of the pages that are featured as well. So here are the data sets by organ system that are currently represented on our site. The largest data sets fall under the endocrine system, as you can see here. We have over 600 phenotypes on the MSKKP, and most of the phenotypes are in the cardiovascular group here. All of our data sets can be accessed via the data tab at the top menu bar, and you can see there's a bunch of different options, so I'll walk you through what we have available. So the first is the list of genetic association data sets, and I'll show you how these results are integrated into the portal in a moment. But this basically shows which data sets are included And if you were to click on the specific link here, it'll take you to more information about that singular data set specifically. We also have data that's tying to transcriptomics and epigenomics, which are available through our sister site, the Common Metabolic Diseases Genome Atlas. So for example, if you were to take this accession number as highlighted here, on the CMDGA, you can actually view the summary for the annotation file that you search. So you can see that these are similar if you want to do a deeper dive into the annotation file set. We also have 253 credible sets that are available on the portal, and these can be explored further in the variant sifter tool. So now that I walked through all of the data, how is this data represented on our pages? So on the homepage, we have a single search as highlighted here, which allows users to search for gene, variant, region, phenotype, or tissue of interest. So I'm going to be searching for the WNT16 gene, and we'll start by exploring our gene page. So here you can see this represents the coding sequence of the gene and the biological role of its product. And this first card that appears shows the functional associations for the gene ontology, and pathways, which are different tabs that you can navigate here. Below this card is the gene level associations, which are based on the Human Genetic Evidence Calculator, the MAGMA analysis, or burden test of rare variants. So this plot has a table below, and further analyses can be done by opening the huge calculator and exploring with this button here. I'm not going to walk through that now, but I just want to highlight that you'll be able to do that in addition to just viewing this plot. So our next card represents tissue-specific gene expression, and as you can see here, this is a box plot with the source data sets, if you were to look here in the evidence, that again link back to the provenance at the CMDGA, which I had shown before, and these are direct links here. This card shows if there were predicted effector gene lists. There are none that are containing the gene that we have represented currently, but they would be represented here. This is a great call. If you have predicted effector gene lists, we could represent those as well for any of the research that's being done. Underneath, we also have the UniProt cross-references and external resources like Ensemble on here. In addition to searching for a gene, you can also search for regions, and if you were to do that, this would take you to the specific region page. Now, this page summarizes genetic associations, credible sets, epigenomic annotations across a selected region. So this page essentially starts with genes that are overlapping in the region, and these that are boxed are protein coding genes, and these are other genes that you can explore. If you were to click on them, the link would take you to their corresponding gene page, which I had previously shown. So this card shows the most significant genetic associations in the region, and the table here shows associations that either have genome-wide significance or are in the top 99th percentile of all associations for that trait across the genome. And again, you can explore in the table, and here you can explore the lead variant, and then also view the LD proxies as well. So the genomic region miner is a locus zoom interactive visualization, and you can explore bottom line variant associations for various phenotypes in this region. Again, I want to point out that if you want to do a deeper dive into exploring the genetic associations, credible sets, and annotations, you can click on the variant sifter button here for that specific phenotype to do a deeper dive into a custom exploration. So here, if you're also interested in searching for a variant of interest, you can explore the variant page, which essentially summarizes the variant impact predictions and genetic associations. This first card shows the closest genes to the variant, and again, if you click on it, it will take you to the gene page. And then beneath this, we have the predicted variant consequences, and the most severe consequence of this variant is highlighted in green here. You can also explore the impact of the consequence based off of the different colors on the impact chart here. This VWAS plot shows the significant associations for the variant across all available traits, and this is generated by our bottom line analysis. And you can see here that this plot is filterable by a few different options, including phenotypes, p-value, and direction of effect. And then the results appear in this table below. Here, the phenotype of interest, you could click and that'll bring you to the phenotype page. And you can also explore the data sets that went into this in addition to the top 25 variants. Lastly, the variant page has transcription factor binding motifs that show altered by the variant. And as I had mentioned, we also have the phenotype page. So here I'm just going to show you what we represent for the example here is osteoporosis. And this represents bottom line meta-analysis, and it accounts for sample overlap. As you can see, the two visualizations that are here are the Manhattan plot and QQ plot, which show the genome-wide single variant associations. And so underneath, this shows the various meta-analysis types for the single variant association signals for the phenotype of interest. So our meta-analysis is this blue color here. And you can see that we've actually identified more associations than just the min-p or largest. And you can see the breakdown of how those are identified here in this value. And again, underneath, we have a table which can help you explore the results shown. We also have the credible sets to cell type method, and this identifies credible sets variants that are located within the regions. And this can suggest which variant genes and tissues are important for specific diseases and traits. Here, again, I just want to point out you can explore more in the variant sifter if you were to click open. Here are the data sets that we used in our bottom line meta-analysis. And again, if you want to look at a specific data set, for example, the Million Veterans Program GWAS Trans Ancestry for 2024, you can click on this and see all the details for that specific data set itself. The Huge Scores tab, which is underneath what I had just shown, show the results for the Human Genetic Evidence Calculator. And essentially, this calculates the weight of evidence supporting involvement of genes in this phenotype. And you can see, you can explore with different filters for genes. And you can also explore the common variant or a rare variant. And if you want to do a little bit more of a deeper dive into what the Huge Calculator is actually doing for their calculation, you can click Huge Calculator Open to explore how the score was decided. We also show the pathway associations that are calculated via MAGMA, and the genetic correlations here, which essentially measure the extent to which two traits share common genetic factors. And so, as you can see here, this is essentially estimated by the LDSC method. And the last thing that we have on the phenotype page is the tissue or cell type specific annotations, which come from CMDGA, as I had mentioned before, our sister resource. And essentially, you can explore through their resource or also explore the tissue page. And these enrichments can suggest which tissues are most relevant for a disease or a trait. We also have a suite of tools that you can explore. And so I'm not going to walk through these specifically, but I just want to highlight what we have available. So first off, we have the Gene Sifter, and this is essentially a tool for finding genes that meet specific customizable criteria. And so basically, you can add in any type of evidence sources that we have available, in addition to expression profiles, to help you find specific genes of interest. The Human Genetic Evidence Calculator, I know I mentioned a few times, but again, it can help you evaluate the extent to which a human genetic evidence supports a hypothesis about the role of a gene or disease in a trait. The predicted effector gene lists basically give you an overview of the genetic architecture of a disease or a trait. The Signal Sifter retrieves variants with a custom pattern of genetic associations. And lastly, the Variant Sifter streamlines variant assessment by combining data and functional annotations to help guide that resource prioritization. So please feel free if you have any questions to let me know, but these are the main pages Like I said, there are a bunch of different tools that are available that can help you guide your research. And with that, I'm going to hand it back over to Noel, who is going to talk a little bit more about the future of the MSKKP and how we're establishing it from what I just showed you to this more established knowledge base. So I'll stop sharing and then take a moment.
Excellent. Thanks, MacKenzie. I thought I saw something in the chat, but where'd it go? Hold on. Eve, what was your question? You have a question? What, you can unmute
I
yourself. Yeah.
EHR, I missed it. What was the definition? I was taking notes.
Electronic
E
health records.
Oh, okay. Thanks, guys. It's like, whoa, what are we talking about?
No
Thanks.
worries. No worries. All right, super.
Yeah.
I'm gonna make sure I leave time for Luke. I don't have a lot, but I just want to set us up. So I had to move outside, so pardon if it's a pretty view, but pardon if I'm a little patchy with my Wi-Fi. You don't want to hear what's going on inside the house. All right, let me get my slides back up. Mhm. Okay. Can everyone see? Beautiful. All right, so I think the MSKKP has three things I can, as you've seen from MacKenzie, it has a heavy GWAS foundation, which is excellent, I think. We also, bring to it meta-analyses that are not done by any other resource already, right? So we take all of the GWAS for a given trait that we can find through GWAS catalog, through literature curation, through direct submissions and contributions, and integrate them and run an analysis that allows us to account for sample overlap, find new loci, and also get rid of ones that may be spurious. So that's already on the MSKKP. So I'll talk about how we're going to expand that. Then, you've got a portal lineage here. What's nice about that, we've been building these for a while. We have a foundation, and a lot of what we do for one common disease portal, for example, CMDKP or the Association of Function Knowledge Portal, we will always put an MSKKP because we think it's very broadly applicable, right? And because of that, we have a small but devoted user base. These people come back again. They're folks who always look up their favorite gene across these traits. So that's great. So that puts us toward, creating a comprehensive resource. But now our next steps are really about depth, breadth, and increasing our usability in new ways. So for that, the proposed resource kind of does this ideally, but not fully yet. I think everything needs to be rounded out, as I just mentioned. Breadth, depth, and usability. But this is really the end game to make it easier for you if you have a list to identify a list of targets for investigation, or to learn what the evidence that supports your favorite gene or phenotype, right? That's really what we're aiming to do here. So the leadership team we've assembled for this next round are folks that may be familiar in our portal universe, but some new folks. Douglas Kill was really, sort of the person who got us here, to be honest with you, in terms of his inspiration and his ideas and his incredible knowledge in this space, right? Actually treating patients and actually being a person who understands these phenotypes better than any of us on the team. Jason will lead up the technical aspects of the platform. Kyle and his team, and you're going to hear from someone on his team right now after me, will head up all the integration of transcriptomics, epigenomics, and functional data. And Michelle and Federico are co-Is who handle different things. Michelle is an expert in translation, and I think she's on the call, so she will help us with integrating other data sets and understanding and running some of our analyses and interpreting the results. And then Federico heads up the IFMRS. So he will bring to us the global footprint, the idea of making sure that we are understanding what are the gaps across musculoskeletal researches internationally, where are the right collaborators to contact and collaborate with, but also how to get the word out about what we're building to more basic scientists, because I think that's where we lack right now. I think we have a good following in the genetic space, but not necessarily beyond that. So that's the team. These are the aims. They're not too shocking. It's very simple. We want to keep it simple and consistent, which is, provide a comprehensive resource for musculoskeletal traits across genetics and genomics. Establish a very transparent and clear governance structure such that we can make these data accessible, fair, findable, and all those things, and more importantly, useful to a broader range of users and ensure sustainability long-term. So we'll break down those aims. Again, this is not surprising. We want to add functional GWAS studies, functional genomic annotations, incorporate model organisms. That's where we definitely lack. And I think musculoskeletal disease research has a strong hold in that area. So bringing that into the resource will be really valuable, we hope. Apply new methods and develop novel ones over the course of the time, both with the research team that I just mentioned, but also more broadly with folks in the space of musculoskeletal disease who are developing methods that would be valuable to the community. So for example, we've already worked with John Kemp, who's part of our early stage knowledge base, to incorporate some mouse knockouts. And that's actually on the MSKKP right now as a labs page that we hope to integrate more formally within the resource. That just gives you an example. Obviously, we want to make this interactive and useful. For aim 2, to do that, to make this governance system, we don't have an external advisory committee right now for the resource, and NIH really does encourage external advisory committees for these knowledge bases, particularly as they go to establish resources, such that you have, cross-domain expertise and disciplines represented on the executive committee. This group will be a guiding body for us, so we're currently recruiting members for that. If you're interested, give me a call. We're going to obviously prioritize, publicize, and make it so that we're soliciting both use case and feedback from our community and investigators, but also, trying to incorporate a range of backgrounds. So the context that you come at musculoskeletal research actually is incorporated in how we present results back to you on the resource. And that's really important to us. So that's really about governance, because it ensures that folks across the spectrum of types of users, how they navigate data, what they want to see, is different. So we want to make sure we're addressing that broadly. And then finally, we want to make sure it's fair and trustworthy. I think this is something that, while we all say we do it, it's important to always keep it in the back of your head to ensure you're doing it well. So to do that, critical metrics. So these are the things we'll be measured by NIAMS and beyond. And this is useful because I think this will make sure that we are, honoring our mission. Again, scientific significance, not a shocker there, but this will be, important to always make sure it's not just a repository, it's just not a knowledge base. It actually is about curation, representation, and integration of results to generate hypotheses or present to you results that may not be available anywhere else. Engagement. We want to make sure that we're creating an impact on the community. So we want to measure that and make sure that we're doing things that actually do make differences. And that will require a lot of, careful solicitation of feedback. And one thing MacKenzie is going to talk about is, how do we gauge how we're doing now? So we're going to talk about what we're doing in that space in a moment. And then quality metrics of, efficiency of operations and service. This is a classic for any knowledge base, not too exciting, so I'm not going to discuss too much about it. And then finally, governance, which I mentioned before. So one other aspect that we find has been a barrier for many people, particularly, not so much in GWAS anymore, but I would say in transcriptomic data and, some of these large scale perturbation studies where there's results that may be relevant to bone and mineral density studies and things like that. There's not an easy way to share data. You may have to do it to like geo and not, for publication, but that's just raw data. How do you create a way for someone who wants to visualize their data alone before it is integrated in the MSKKP? Or for publication, they want to make it accessible, like it's a particular analysis that they've done, and they want to make it useful, but they also want to contribute it. So one of the key components of the Musculoskeletal Portal over the next 4 years is to create a registry system that any type of data that folks want, I'll share that data list in a minute, data type list in a minute, can submit it. It's easy, it's free, you control all of the information until you're ready to share it, meaning like it's all protected for you, and we can QC it with you. We can provide a dashboard for you to do things like QC, meta-analysis of just your own data. You can also upload your own annotation and see how it looks against all of our other annotations and methods. But we don't do this as an incentive for people to share their data to increase that resource so that it's a broader nurturing activity for the broader community. So that's one thing you'll see come out of this. It will, not only benefit musculoskeletal disease community, but also our entire common disease research community. So the types of annotations, I'm not going to go through this in huge detail, but they're not, uncommon to some of the things we've been doing in the Association to Function Knowledge Portal, but these are data types that range everything from, GWAS, but also to molecular QTLs and perturbations, cellular readouts and screens. All of these information are generated in many cases for musculoskeletal disease, but not in one place, as I mentioned, standardized and represented. Even from what I gather, in doing and applying for this one, we actually looked through many resources and realized even for like some of the canonical resources, they're light in the tissues that are relevant to musculoskeletal disease. So a couple things we plan to do. Add trait coverage. We're a little light in things like sarcopenia and muscle traits, so we'll be beefing that up over time. We also have the benefit of adding some longitudinal imaging phenotypes. These are really quantitative traits that help measure things like cartilage thickness and T2 mapping that are important in diagnosing, OA and other musculoskeletal related traits. So what's great about that is we can work with other resources that have that information, incorporate that here, and harmonize the results of that onto the portal. We can incorporate cross-species analyses. So from mouse to human is one direction we plan to go, and surface that ontology. So obviously there's natural connections between those two resources, such that if you see a mouse knockout, we can actually map it to a human phenotype using genetics, not just semantic similarity. So that would be a nice added bonus on the portal. And then embed things like, readouts like DEXA and things like that. This could attract a slightly different user base, people who work in the orthopedic section, because these other metrics may be useful in understanding musculoskeletal disease from a holistic perspective. So that's about GWAS traits, and we have a lot of data there, and we plan to do that exactly how we've done before, with GWAS Catalog being our partners in crime when it comes to GWAS data sharing. And that will be something, and also our contributors, because those are the greatest people we've worked with to bring data into the MSKKP. So tissues. This is where we have a lot of work to do. And thanks to Luca, who you're going to hear him through a minute, and others, we hope to focus on 5 key tissues where data are available, but also hopefully, as data come online. Bone, clearly, but it's a hard tissue, right? So you've got things like joints, synovium, skeletal muscle, tendon and ligament, and intervertebral discs. So these tissues are not currently represented very holistically within the portal. So we're hoping to engage, basically searching public resources, collaborators, our community, and literature curation to incorporate what's currently available, particularly from canonical resources when they are available, but also directly from contributors who are generating these data. And we're going to start with bulk RNA-seq and single-cell RNA-seq. We do this because I think it's been something our users have wanted. Some of that data is already represented on other portals, so we have a means to incorporate it. But creating integrated maps for these tissues would be, I think, a value add to the MSKKP. So we're going to hear about that. Just really quick before I stop and hand it over to Luca, I think other things that we want to do that are a little exciting, some future-facing analyses will allow you to do causal inference mapping. I think that's a goal for us over the coming years. We want to sort of use the infrastructure that many of you heard about related to Pigeon, which is the extension of the huge calculator, as an infrastructure to determine latent mechanisms in different musculoskeletal data sets. So this will give us like pathways, things like that have already been discovered, like the Wnt signaling pathway, for example. Other things like, incorporating agentic AI into the resource will come online with that, allowing you to have snapshots so you can actually download and access the results we've done, but also navigate through LLM-ready representations of the results in the MSKKP. And again, we want to make it so anybody who contributes gets incentives. You are the most important components of the resource that we built. So we want to make sure we have really cool incentives for that. And finally, fairness. This is, again, something we just don't want to take lightly. And also in a world, just one more point about this, in a world where there's so many resources right now, I think, especially in common disease, we have been collaborating with a lot of things like these common fund resources, which are, more disease agnostic activities, right? But to do that for all of common disease, those common diseases need curated resources. So in order to bring musculoskeletal disease research up to the level where it can readily share its results with these broader, data fabrics that you're hearing about, and NIH-wide activities to incorporate data, we have to bring the MSKKP sort of up to that level of other disease areas, for example. So that's a take-home point that I want to really make. We're going to go, many resources are deep and narrow. We want to, round out the resource over the coming months and years. And we really hope that if we kind of fuse the rigor of those lovely Mendelian databases that many of you use. They're really beautiful with our existing knowledge portal infrastructure for scaling of GWAS and layering of multi-omics data. We really hope this will be the backbone, if you will, not to be too cheeky, for musculoskeletal genomics. So that's kind of my final thought. And with that, I want to show you from Luca. Luca's going to show you some of the work that we plan to do in the space of aggregating more tissues and incorporating those into the portal for you to interrogate along with the human genetics. Did I miss any questions as we go? As I stop sharing or comments? Okay.
Eva?
Eve, what's
Okay,
up?
sorry. Basically, that there's two functions now on Zoom. One is chat and one is questions. The questions uh portion is great, but the chat, some kind of AI thing is interrupting. And the next thing is I don't have the ability to quickly listen and give you feedback, so I'm taking notes. But one of the most important things I think in the world today is for people to be comfortable with AI because it's scary and we need to kind of understand a little bit better how it works and how we might use it more effectively.
Absolutely. I completely agree with you. I think one of our visions is, sort of like bringing AI, the technology, into, not to use the enclave of our data. And that combination, I think, if you are aware of that as a user, like you are only playing in this playground, for example, like integrated GWAS data, transcriptomics, and analyses that have been done on it and results. If you knew that that was your constrained environment, we want to make it so you can ask questions rather than having to necessarily navigate through a bunch of pages. Sometimes you're like, I don't want to see all these pages. It's too much. I just want to know, what is the evidence that supports my favorite gene? Wouldn't it be nice if you could say, I'm going to come to MSKKP and if I like chat windows, which you could have, you could be prompted in a set of steps to say, with this orientation, you give us a little bit of information. You are you are a retired medical researcher, you are an RN, CDE, blah, blah, blah. You're interested in, in metabolic disease. You type in that information. And then under that model, you'd get back results from just the prescribed infrastructure that we've created. So that gives it the trustworthiness. Does that make sense? You're not hallucinating by searching the entire web. It's allowed
Right,
to
right.
have that constraint. Now that's not going to do everything perfect, but the constraining the searching to the data is, I think, a first step at making it something useful for our field. That's just
Right.
my two
And
cents.
I can't fit everything about myself on a Zoom. I started working with computerized accounting in 1973. So
Yeah.
I'm not going to be your average user. I've tried to keep up with relationship programming and so forth. And that I guess is what I'm trying to figure out. And let me think about it for a while and maybe send you a picture.
Sure.
Does that make sense? Because I'm a big picture person on
I
how to use the tool so that like not me, but a person across the street from me that doesn't even have a bachelor's degree might be able to ask a question, and it might take you like a week to really answer it in terms of the genes and
Mhm.
variants for her, but she would have a comfort level that your information is correct.
Yeah.
Right now we have Grok, answering all kinds of stuff
Yeah.
on X, and I have Bing irritating me because it wants to tell me something I don't want to know when I just want something else. You get me.
Definitely.
So anyway, thank you. Yeah, and let me think about it for a while and send pictures.
No
Is that
problem. That'd be
okay?
great.
Oh
Beautiful.
yeah. I mean, we've been together a while now, since 2020.
Yep. Happy
So
to.
thank you.
You're welcome. All right. So, Luca, why don't you tell us a little bit about some single cell maps, please?
Yeah, love to. Okay. Can you guys see my screen?
Yes.
Cool. It seems like you can also hear my voice, which is great. Okay. I'm Luca Tucciarone. I'm a fifth-year postdoc in Cal Galton's lab. Our team is mainly involved in the back end of everything that Noel and MacKenzie were talking about. So me, Kyle, Yuna, Sarah, Sierra, Liza, Parul, and Ying are the main actors in the back end of MSKKP and other portals that are related to our lab. In particular, we specialize in creating single cell maps. And this is an example of a single cell map that a very talented PhD student in our lab, Weston Ellison, made for a project that is very adjacent to MSKKP. We were able to integrate 2.2 million cells across a number of modalities, data sets, hundreds and hundreds of donors, and layered disease status. And the vision of what we would like to do for MSKKP and other portals that are related to this is to be able to reach this level of cell atlases as Noah was talking about, like NHA level atlases that people can use as a resource to look for their favorite gene expression or peak sets in their favorite cell type in a particular disease state. The workflow that we have set up for doing so is actually pretty simple. And the most difficult part is collecting publicly available data sets that are ready to be processed. So I will use this as an opportunity to empower everyone that has a data set that would like to be featured in MSKKP or other portals that are adjacent to MSKKP. Please, I will implore you, reach out to us. And if you give us the process data set, we swear we're going to use it and we're going to make good use of it. Because that is by far the most difficult part of this whole process, is collecting trustworthy and publicly available data sets with metadata that's relevant to the at the donor level that we can use for this portal. So we collect all these public data sets and we integrate them together by a number of steps, by a process that we can summarize as reference mapping, where we basically take the biggest and most comprehensive data set and we use it as a reference to project cell type annotations of all the other data sets that are a little bit smaller and out there. Then we integrate all of those cells together in one big map, and then we can create downstream assets. Those downstream assets will be then used in MSKKP with their beautiful visualizations. They can be UMAP with layered cell type annotations. As of now, we are working on 3 layers of cell type annotation. Which are cell lineage, lymphoid cells, cell type, T cells, cell subtypes, CD4, CD8 T cells. Along with that, we plan to release metadata table at the donor level and at the cell type level and at the cell level, count matrices based on the type of data that is available for this particular for a particular tissue. You may have different types of count matrices for attack and RNA-seq, for example, differential expression and differential accessibility based on disease status, peak set, and cell-cell communication outputs in terms of ligand receptor pairs across all cell types and different disease states, or even metabolite sensors type of analysis where we are able to infer metabolites expression based on the expression of the enzymes and genes that are related to their presence in the cell. The resources that we have been using up to now, it's a number. We have been scraping data from HubMap, SelectGene, UCSC Data Portal, Human Cell Atlas, Azimuth, Broad Single Cell Portal, and publications. Which brings me back to the point that I was doing before. As long as the data is present on a portal, it's pretty easy to download, but when it's a publication-related asset, it gets kind of complicated to grab your hands on the full set of analysis and metadata. So please reach out to us. This is a pilot integrated map that we actually already performed for skeletal muscle, one of the 7 tissues that will be featured in the initial release of MSKKP. We have integrated a number of data sets and almost 400,000 single cells. We have two different layers of cell type annotation at this stage: cell types and cell subtypes. As you can see, smooth muscle cells, satellite cells, and muscle fibers. Among them, muscle fibers can be split in based on their type, and so forth. So you can see the number of cells and the proportions of those cells in the pie chart. And also on the left side, you can see the metadata level analysis, where we can list the number of cells per dataset, how many donors were present in that dataset from a heat map, if donors were control, unknown, or disease, the overall age of those donors, if we can find males and females, and like based on like again the metadata that is available in the dataset. As of now, this is the one map that we were able to generate for MSKKP. We have already started to look for the other tissues. We found a couple of data sets for bone. Those are mainly publication that we can find in PubMed. We found 10 different maps that are available for tendon and ligaments. As you can see here, like most of them will require us to connect with the corresponding others to get the process data. 13 different data sets from HubMap that we can use for cartilage, mainly in the meniscus area. 5 different data sets for intervertebral discs. 6 for synovium. 9 for bone marrow. And that would be it. So across all these maps, we aim to create single cell maps that will contain hundreds of thousands of single cells to generate those assets that I've listed before as a resource for everyone to use in conjunction with all the other downstream analysis that Noel and MacKenzie were talking about. Thank you so much for listening.
Thanks, Luca. I'm just going to pause before I go into my final slides. Does anyone have any questions for Luca that you have? Also, we can take questions after as well if you want to think about it a little bit. Okay, I think we'll just head on to the next section. Thank you again, Luca. And you know, after we had just heard from both Luca and Noel, I want to talk a little bit specifically about how we're planning to mature the MSKKP in terms of our feature and what could be coming. And so if you would like to help us shape the MSKKP, please feel free to provide feedback in this survey. I'm just going to walk through what we've already started to gather, and if there's anything else that you feel really passionate about, we'd be really eager to have your feedback. And so the initial results from the survey show that most users are from the US, the UK, France, Italy, Germany, and Japan. And they are mostly part of IFMRS Society, but there are also members from the American Society for Bone and Mineral Research, the Japanese Society for Bone and Mineral Research, and the International Bone Marrow Adipose Society. And so when asked what types of data should be included in the MSKKP, we saw that most people were asking specifically for omics data, which is great because, as Luca has already shared, we are already making headway in this space. For the genetic data, people are interested in, you know, obviously the GWAS, but also the pathogenicity of a major or rare gene variants. And then one aspect that we don't fully have yet in any of our knowledge portals, but something that we're very interested in is clinical and imaging data. And obviously, this data should be high quality, and you know, hopefully we would have reliable data set curation as part of that. And as Noel mentioned with the data registry, I'll walk you through what that would look like as an effort to hit that type of data quality. Other knowledge and visualizations that were of interest included cell type specific gene expression patterns and regulatory networks tying as the two most wanted. But I just want to note that people are also interested in pathways, interactions, and experimental data. For which phenotypes or traits would you like to most see represented on the musculoskeletal portal? There is a list that we've already been thinking about, as Noel had previously mentioned, but survey responders have indicated anything regarding bone health, so specifically fractures, bone mineral density, osteopenia, and osteoporosis. For skeletal disorders, OA, adolescent idiopathic scoliosis, OPLL, lumbar disc herniation, idiopathic necrosis of the femoral head are a couple others as well. People are also interested in rare bone diseases, anthropometrics, and obviously genetics. And so one thing that we really care about as we're building this portal out is how could the MSKKP change how you're doing your research specifically. And so people are saying that the data organization and analysis would be extremely useful if there was an expert who could help us figure out common criteria to score and organize this data. And I think some of this will come through, you know, those who are helping us shape the portal with this feedback, but also our external advisory committee, as Noel had mentioned previously as well. And having that shared system would essentially make it more useful and reliable in the long term. Having the ability to do a easy, broad meta-analysis crossing data, study, and format is highly requested as well. And also high organ diversity, so if people could look at organs that are other than just bone. And then, as you can see here, people have indicated that there is a desire for other animal models in addition to human that are most relevant to research. And as you can see here, mouse is highly of interest. And as Noel had mentioned, we are working with John Kemp in this space for, you know, the mice. And obviously there's the zebrafish and rat that are of interest as well. So this is something that we'd like to include. And one thing that Noel has mentioned is the desire for us to include a way to implement data and, you know, gather this data and represent it on the resource, but also give you, the researcher, the opportunity to have a closed environment which allows you to register specific data sets and run, you know, QC and meta-analysis. This is something that we have for a beta resource for the Hermes Consortium. And so essentially here, this is the main dashboard and basically shows the data sets that have been added specifically by users and what status they're in. So whether they're approved, ready for review, whether they're submitted to QC, and if they've failed QC. And so I'll kind of walk through the various pages that we have here. But here you can see users can upload GWAS data and undergo this quality control by submitting this workflow. Essentially, there's a couple different steps. Once you enter the metadata, you would then upload the file, you would map the columns, and then this data would be available to upload. And so as you're walking through this, it basically gives you prompts on what you should be including for this data upload. And if there's a require field, it'll make it known. So it has all the information right there for you as you're going through the data registry. Now, once the data has been uploaded, as I had mentioned, users can run QCs. And so if they have the designator approval role, they can actually determine whether or not this data set passes the QC checks. So you can see here, there's a whole report. And if you scroll down, you can look at various QC metrics and also like QQ plots and things like that as well that can help you. And the QC method can be, you know, useful, again, if you're just looking at the data quickly and comparing it to what we have in the resource. And then here, Noel had also mentioned you can run some meta-analyses. And so these are just a couple different examples of, you know, combining various data sets and seeing, you know, what the results would look like from there. So with that, these are just some things that we're thinking about in terms of the future of the MSKKP. But if you're interested in helping us shape the MSKKP directly, you can contact us at help@kp4cd.org, and that'll go directly to me. So I will be able to see all of these requests, and we'll be able to communicate those with our engineers. And also, if you are interested, please fill out the feedback survey. We are more than happy to gather any and all information. And so while the survey has a couple more like structured questions, I just showed you a very high level of them. It is more in depth. So if there is something specific that you would like to discuss for feedback that's not represented on that survey, please feel free to reach out to me. So with that, thank you everyone so much for taking the time. Thank you, Luca, for giving us an overview of the single cell data that's coming and the mappings. So I know we have a few minutes left, so if anyone has any questions, please feel free. We have the time to do so. But we're really looking forward to expanding this out and really have the user as the main priority when shaping this through.